So, a bit of caution for budding young evolutionary biologists. Superficial appearances can be greatly misleading. So, you remember the garter snake and the glass lizard? Well, consider these two lineages: a bottlenose dolphin on top and a great white shark on the bottom. Morphologically, they look very, very similar. Both species have this streamlined torpedo shape so they can move fast through the open ocean. Very, very similar fins. They have a dorsal fin on top and pectoral fins down below. If you look closely, they're even kind of colored the same way. So both species are going to have what's known as counter shading, where they are darker on the top and then lighter on the bottom. So this is going to camouflage them both from above and below, from both predators and prey. Both of these are clearly very effective open water hunters. So they probably share a pretty recent common ancestor, like grizzly bears and polar bears, or gray wolves and coyotes, right? Or at the very least, they're probably in the same family, like all of the cats, which look morphologically kind of even more distinct than these two look. Not even close. So the dolphin, a cetacean, is a mammal. Like you and me, it's got lungs, it's endothermic, it gives birth to live young. The shark down below is a cartilaginous fish with gills. The shark's ancestors never lived on terra firma, on dry land, like the dolphin's ancestors did. That dolphin shares much more genetic material with you and I than it does with that shark. So, this phenomenon is known as convergent evolution. This is when two independent lineages on the tree of life converge on a similar form, on a similar morphology. Oftentimes, it happens, just as we've discussed, when the lineages inhabit similar ecological niches. And we're going to talk more about the ecological niche, where the organism fits into the ecosystem, later in the semester. But essentially, these two organisms, they make their living the same way, hunting fish and other marine organisms in the open ocean. So, counter shading, for example, this evolved independently in both of these lineages, because it doesn't matter if you're a mammal or a cartilaginous fish, that color pattern is going to provide a selective advantage in the ocean where these two lineages hunt. So, this is a broad phylogenetic tree of the vertebrates. And if we look right here, this is the great white shark. And way over here is the dolphin, right here, sharing a common ancestor with the other whales, and then the artiodactyls, the herbivores. Again, the common ancestor of the dolphin and the human, much closer than the common ancestor of the dolphin and the great white shark, which here the white shark is related to all of these cartilaginous fishes. So the dolphin, much more closely related even to reptiles, the class aves, the birds, even the lobe-finned fishes, much more closely related than to the shark. In fact, you have to go back over 400 million years before you find a common ancestor for the great white shark and the dolphin. All right, an important distinction. I want you to get this straight in your heads moving forwards. A homologous structure, this is something we've discussed in the Evidence for Evolution lab. Homologous structures are those structures that share a similar embryonic origin, meaning they are derived from similar genes and similar developmental pathways. So, for example, the fin of the whale and the arm of the human are homologous structures. All vertebrate limbs are homologous. They share similar embryonic origins. They all consist of this basic architecture with a humerus, a radius, and an ulna, metacarpals, and phalanges. So these are homologous organs. Okay, another important term, an analogous organ is one that has a similar function. Okay, so the pectoral fins of great white sharks and dolphins, those perform a similar function. Even though they have different embryonic origins, those are considered analogs. The human arm and the whale flipper, although they're homologous in their origin, they are analogous in their function. They do different things, right? My arms are for waving around while I'm recording videos, whereas the whale's arm is evolved into a fin. All right, to bring this point home, the eastern bluebird, the pterosaurs, which of course are an extinct lineage of flying reptiles, and the Samoan flying fox, which is a fruit bat, a mammal, a flying mammal. All three of these vertebrates, their limbs are homologous in that the bones that they have all develop from similar embryonic origins and go through similar developmental pathways. They all have the same architecture: the humerus, the radius, and the ulna, the metacarpals, and the phalanges. Why? Because they inherited this architecture from a vertebrate akin to Tiktaalik with that architecture. We know that the wings are analogs in that they're all doing the same thing, but flight itself is analogous in these vertebrate lineages, meaning that flight evolved independently at least 3 times in the vertebrate lineages. And how do we know? The different vertebrate flyers are doing it differently. So the bones are morphed into very different shapes and sizes in the bird, the pterosaur, and the bat. So, for example, look at the pterosaur. It still has grasping fingers. Only one finger is really elongated here, whereas the bat, all of the phalanges are totally elongated. Okay, so flight evolves independently. Flight itself, the modification of these homologous organs, occurs independently at different points. And then to compare that to insect, because we know that flight evolved in the insects, these, again, they're analogs. They're both used for the same thing, but these are definitely, insect wings and vertebrate wings are not homologous. They share different embryonic origins, different DNA sequences. So hopefully that helps. To further help you understand the difference between homology and convergent evolution, your lab this week is really wicked cool. So you're going to be taking a trip to the Caribbean and you're going to be exploring anole evolution across a variety of islands in the Caribbean. So when we look across islands in the Caribbean, we see anoles that have similar ecomorphs in that their morphology is very similar and they inhabit similar ecological niches. So, for example, in Puerto Rico and in Jamaica, we see different species of anoles. These anoles are not capable of interbreeding. They're different species, but they have very, very similar morphologies and they have very, very similar ecological niches. These large green anoles live way up in the tree canopy. Similarly, on Cuba and Puerto Rico, we find this twig specialist. So these different species of anoles have teeny weeny little arms, and they are adapted for clinging to and hunting on very tiny twigs with small diameters. Grass specialists, like the species found on Hispaniola and Cuba, these guys have really long legs and really long tails. They're sprinters. They're very, very good runners. So the question that you're going to be asking yourself is, so take the twig specialists. Do these two species look so similar morphologically and inhabit the same ecological niches because they share a recent common ancestor, and this morphology is homologous to that ancestor? Or have these two lineages evolved into this morphology independently on both Cuba and Puerto Rico? And the similar structures are simply analogs. They do not share a similar homologous origin from a recent common ancestor. So the question is, how do you do this? To tease out these relationships, you're going to use DNA sequences to examine molecular homologies, the amount of shared DNA between the different lizards. And from these molecular homologies, you're going to build phylogenetic trees. And ultimately, you're going to understand why islands are such amazing evolutionary laboratories.